The influenza pandemic of 1918 killed somewhere between 20 and 75 million people and infected roughly one-fifth of the world's population at that time. It infected 28% of all Americans, and an estimated 675,000 Americans died of influenza during the pandemic. In this presentation, we will explain the qualities that make up the influenza virus's virulence and lethality, and how it relates to the 1918 pandemic. There are three types of influenza viruses, A, B, and C. In this presentation, we will focus on the influenza A virus. The B and C type viruses are less complex versions of the A type viruses in that they have less genes. The influenza virus contains 8 pieces of RNA in its center, surrounded by a protein structure called the capsid. Outside of the capsid is the lipid envelope. Attached to it are the neuraminidase and hemagglutinin. The hemagglutinin is attached to the cell's receptor sites and gains access into the cell, and the neuraminidase plays a role in the virus's escape. There are 13 types of hemagglutinin and 9 types of neuraminidase. So when you hear the name H1N1, it refers to the first hemagglutinin type and the first neuraminidase type. This means that H1N1 swine flu and the H1N1 Spanish flu are essentially the same virus. Hemagglutinin is the protein that attaches the virus to the cell, but it likes to bond to cell receptor sites in a very specific way. A virus that infects animals must change its hemagglutinin structure in order to attach to human cells. Cells use endocytosis to absorb molecules on their surface by engulfing them. It is used by all cells of the body because most substances important to them are large polar molecules that cannot pass through the hydrophobic plasma or cell membrane. The virus enters the cell by tricking it into thinking it is an essential protein and enters the cell covered in the cell's membrane. The cell releases a lysosome, which is normally used to break down proteins, and it merges with the endosome containing the virus. The acidity of the lysosome releases the virus's RNA packets, which are specialized to locate the nucleus and enter it. Once inside the nucleus, the RNA then gets to work replicating itself and gets the cell to start making the proteins needed for more viruses. The ingredients for a new virus collects at the cell membrane. The virus then pushes itself out, taking a part of the cell membrane with it. The hemagglutinin attaches back to the same cell receptor site on the membrane, but the heroic neuraminidase finally plays its role and acts as a pair of scissors to snip away the bond so the virus is free to go and infect other cells. The virus is spread from organism to organism through contact with contaminated surfaces and ingestion of sneeze and cough droplets.
So why aren't we all dead? These viruses sound pretty specialized and efficient, but don't worry because we have an immune system. Our white blood cells patrol our bloodstream searching for infections, and when they find one, the immune system is alerted. Immune cells are sent to infected sites, and antibodies are released that latch onto viruses and interfere with key processes, like the neuraminidase ability to cut newly formed viruses loose. They also clump viruses together to be eaten. Specific sets of antibodies remain in your systems in case the infection ever returns. For unknown reasons, your immune system can occasionally flip out with certain infections. A healthy immune system will produce and send large amounts of immune cells, and the increased concentrations can cause inflammation which interferes with respiration. In the case of the 1918 pandemic, if you had a healthy immune system, then it was far more likely for your own immune system to kill you in the case of an infection than it was for the virus to kill you. This is the reason for this W-shaped graph, because those with the healthiest immune systems were more likely to die from their own immune system. The death rate for the 15 to 34-year-olds of influenza and pneumonia were 20 times higher in 1918 than in previous years. So why do we keep getting infected? There are two processes that change viruses, antigenic drift and antigenic shift. The first is basically the same way human cells change. Over time, errors in replication cause mutations, and then, when the mutations accumulate, large-scale changes in proteins and processes occur. In the virus's case, the shape of the hemagglutinin and the neuraminidase can change so that antibodies can no longer stick to them. Antigenic shift is when an animal is infected by two types of viruses, which results in a completely new combination of hemagglutinin and neuraminidase. This was the case in the 2009 swine flu, when presumably an avian flu and a human flu combined in a pig. At the moment, there is limited information on exactly how the 1918 influenza virus works. The virus was being recreated from a frozen sample so that research could be done to halt further pandemics, but research has recently come to a halt due to a moratorium on experimenting with these deadly viruses for fear of it escaping the laboratory or being used for bioterrorism. It is important for the further study of these viruses because if it ever happens again, we will be helpless because of the lack of published information. In summary, if there is another pandemic, then the scientists won't save you and your own immune system will probably kill you. Go figure.